Hi, my name is Emily Ray, and I'm a first-year PhD student in Karen Maruska's lab at Louisiana State University. Today, I'll be presenting my work on the sensory and neural mechanisms of parent-offspring recognition in a maternal mouth-brooding cichlid. Parental care evolved independently several times and is present in a wide range of species. While parents are providing care, parents and offspring can recognize each other using auditory, chemosensory, or visual recognition. Parent-offspring recognition can also be multimodal, meaning that more than one of these signaling modalities is involved in recognition. The maternal mouth-brooding African cichlid fish Astatotiliapia burtoni is a particularly interesting model to investigate the sensory and neural mechanisms of parent-offspring recognition. Females brood developing young in their mouths for about 14 days and then provide post-release maternal care by protecting young in their mouth when threatened, as you can see in this video. Because adult A. burtoni cannibalize fry, parent-offspring recognition becomes critical during the post-release maternal care phase. Mothers must be able to recognize their fry to avoid maladaptively consuming their own offspring, and fry must recognize their mothers to avoid swimming towards an unfriendly mouth when threatened. Using this system, I aim to determine which sensory signals drive parent-offspring recognition and to identify the neural substrates of parent-offspring recognition in A. burtoni. To answer these questions, I set up a behavior experiment in which females are exposed to different sets of stimuli from their fry. Control females receive no stimuli from their fry. Some females receive only visual stimuli from their fry. Some females receive only chemosensory stimuli from their fry. And some females receive both visual and chemosensory stimuli from their fry. I record the females' behavior in response to these stimuli and analyze their behavior using taxa, a free open-source animal tracking software. Using taxa, I compared the average speed of the females in each treatment group over the 30-minute trial. Control females are shown in red. Females that received only chemosensory stimuli are shown in green, females that received only visual stimuli are shown in blue, and females that received both chemosensory and visual stimuli are shown in purple. Females that received only one stimulus, whether that's chemosensory or visual, swam faster over the 30-minute trial than females that received no or both stimuli. Another way to look at this data is to compare the total distance swam, where we see the same trend. So females receiving only one stimulus swam further than females receiving no stimuli or both stimuli. While these differences aren't significant and the data are still preliminary, if we look at the tracks swam by individuals in each treatment group, we can begin to see why this trend has emerged. To orient you to this picture, it was taken looking down into the tank where behavior trials were conducted, and the red bar denotes the site of stimulus delivery. The green line shows everywhere a female swam during the trial, and this track is from a control female, so a female that received no stimuli from her fry. You can see that she swam throughout the tank, but didn't spend much time swimming in any one area. If we compare that with tracks from fish that received only chemosensory or only visual stimuli from their fry, we can see that these fish swam much more than the control fish, but again, the swimming isn't too concentrated in any one area. Finally, if we compare these tracks with the track of a fish receiving both chemosensory and visual stimuli, we can see that she swims less than the fish that received only one stimulus and spends most of her time swimming in one area, which is the site of stimulus delivery. While this data is preliminary, it suggests that both chemosensory and visual stimuli from fry may be necessary for A. burtoni females to correctly identify and locate their fry. Following behavior trials, I collected brains from focal females and performed antibody staining for PS6, which is a ribosomal protein and marker of neural activation. This graph depicts the PS6 cells per square micrometer in the DC5 region of the central dorsal telencephalon. Females that received both chemosensory and visual stimuli from their fry, which are shown by the purple bar, have more PS6 stained cells per square micrometer than control females who received no stimuli from their fry and are shown by the red bar. This is in line with previous results from the Maruska lab that showed an increase in PS6 stained cells in the DC5 region of females providing maternal care compared to females that were fed or starved. Next, I compared the number of PS6 stained cells in the DC4 region of the central dorsal telencephalon. Here, females that received both chemosensory and visual stimuli, again shown in purple, had more PS6 stained cells per square micrometer than females that received no stimuli, only chemosensory stimuli, or only visual stimuli. While there isn't yet a clear consensus on the mammalian homologue for the central dorsal telencephalon, some evidence suggests it could be homologous to the mammalian neocortex. The mammalian neocortex is involved in processes such as motor control and sensory processing, both of which would be critical for offspring recognition and location. So to summarize the work I presented today, my preliminary results suggest that A. burtoni females may rely on both visual and chemosensory stimuli from fry for offspring recognition, and that different stimuli from fry result in different neural activation patterns in A. burtoni mothers. As I continue this work, I'll also be exposing fry to the same suite of stimuli from their mothers to determine if parent-offspring recognition is bidirectional or unidirectional in A. burtoni. Because fishes are the largest and most diverse group of vertebrates, identifying the sensory and neural mechanisms of parent-offspring recognition in a model fish species will give us insight into the evolution of parent-offspring recognition across vertebrates. Further, as freshwater environments are rapidly changing, understanding which sensory signals are used in parent-offspring recognition will give us insight into how species may adapt to changing environmental conditions such as increasing turbidity. With that, I'd like to thank the Maruska Lab, especially Marie Drozda, an undergraduate who's been helping with this project, and I'm happy to take any questions.